on this computer. Fantastic. Okay, so that is the very first thing that I want you to know about. This is Chem 1330. One of the great things about an online class is that it's really tough to go into the wrong place, because we've got the barrier of I only advertise this link on our Canvas page. So at any rate, that's what we got. Um, I am Dr. Gray. I will be your instructor for this class this semester. This is a second semester chemistry course. Many of y'all, or I believe absolutely every single one of you, took Chem 1310. I suppose there's a possibility somebody did not, in which case they had some sort of other transfer credit or something like that. But this is a second semester class, and if I remember correctly, every single person in here is a non-science major. So your communications or a business or I don't think ag is in the College of Science, but like a design major of some sort. So you're looking to get a second science credit to fulfill the requirements of your degree. This is, I think, kind of a cool class because of the fact that you have a little bit of free reign, in the sense that in a second semester general chemistry class, you're doing a lot that's different from what we're going to be doing because we don't have to take a standardized final exam. I'll be writing all of our exams and they're tailored just for our class. So there's no national or international exam to compare to. Now, today we'll talk about kind of the nuts and bolts of this class and how it functions and everything like that. But we'll also talk a little bit about what we're going to be covering this semester. So the first thing that I wanted to open up was, and just so everybody knows the how to navigate and stuff in this class, if you have any questions, feel free to do the hand raise function or just speak up, or you can use the chat function. I do my best to monitor the chat, but sometimes I lose sight of it. So the first thing that I've got popped up or pulled up is I have our syllabus. Now, I think when it comes to the syllabus, there's a lot of it that's just kind of, it's very similar to any other class that you've taken. I like to keep my syllabus as succinct as possible, and you'll see that on this syllabus. It is pretty simple. You don't have to buy anything for this class. The materials that we'll use, I will either provide from Open Education Resources or OERs. Sometimes we'll use this textbook, this Chem Libre text map link that you have here. It's Chemistry for Changing Times. If you took my Chem 1310 class, you'll use the same thing. Okay. Yes, the syllabus. I'm sorry for the syllabus is up on Canvas. And let me go ahead and open our Canvas page. I believe that I just made some changes to it, so it should be up to date now. But let me go ahead and pull up Chem 1330. You know what? Okay, just 1 second. Use this the front page, and then pages front page. Got it. Okay, so now I'm going to switch my share to our Canvas page. This is our Canvas page. This is how I have it set up and how I manage Canvas pages. Typically, I just like to make a simple table. And I like to sum it up as much as possible. Week 1, day, date, and then what was on the agenda for that day. I post any of your lecture notes as both a PDF and as a PowerPoint file. The syllabus is accessible here as a PDF. And so you'll see that there. Okay, then let me go ahead and switch over though. I did something strange to my note. Go back there. There we go. So Now what I want to do is I want to switch over to, well, no, I don't want to do that. Okay. When you're looking at our Canvas page, what you'll see is that we've got a couple of exams. Our first exam is going to be September 17th. Our second exam is October 8th. Third is October 29th. Fourth is November 19th. And our fifth exam is December 5th. So that means we've got a total of 5 exams, and we also have a final exam, which is administered on the 12th of December, from 2 to 4:30 p.m. So that's kind of the nuts and bolts of important dates that you need to know. Periodically throughout this semester, we'll be doing group activities and class activities, and I'll have those noted on our syllabus or on our Canvas page. One thing that I do always like to do is December 12th. In the event that you ever need to look up when your exams are, a good resource is to be able to go to the registrar's homepage and pull up the academic calendar. This is our academic calendar for the fall, and it's always confusing because the very first date is actually the spring of 2024. But if you scroll through, that's because that's a registration date. If you scroll all the way down, you'll find, okay, final exams for our full term, those begin on December 6th. There you can click final exam schedule. When you click final exam schedule, you will see these different options. Click fall 2024. Scroll through here, and this sorts them based on the meeting days. Our class meets on Tuesdays and Thursdays. So we're looking for a TR, and then we're looking for 2 to 3:20 p.m. So there's our 2 to 3:20 p.m. Thursday, December 12th, 2024, 2 to 4:30 p.m. So I always like to do that just to confirm that I have the right date on our Canvas page. So I've got the right date there, so that's good to know. Now what I'm going to do, so that's all available to you. I'm going to stop my share. Now I'm going to switch my share. Now I'm going to switch over to a PowerPoint presentation. So sure. Okay. This is Chem 1330. I'm Dr. Gray. Welcome to this class. Welcome to our class. This is a picture of my family after we floated the river. I've got 3 little boys there. This is my youngest son, Arlo, A-R-L-O. This is he is 4. This is Walter, which at a very young age, we said, oh, we're going to call him Walt. And like immediately he said, my name is Walter. And so we were like, we're not going to call him Walt. He is 6 and he's going in 1st grade. Then this right here is Mr. Otis. And he is 8 years old as of last Friday, and he's going to be in 3rd grade. This is my wonderful wife, Catherine, and she has graduated from school. So she's not in school anymore. Likewise, well, I kind of haven't because I obviously kind of am still in school because I work at a school. But this is us. These are the most important people to me. Now, who am I? I was born in this wonderful state. Does anyone know what state that is? I'll give you a hint. It's not West Virginia. So given that it's not West Virginia or Texas, you have 48 other options. It's not Alaska. It's 47. It's not Alabama. Illinois is close. It's not Illinois, though. It's not Michigan. Also close. You're getting very close. Wisconsin is the correct answer. Wisconsin, where Houston Texans legend J.J. Watt is from. I was born in Wisconsin, as was J.J. Watt. So we're kind of family in some respects. So, oh, that's, yeah, there we go. Wisconsin. I was born here in Milwaukee County, and then around the time I was about a year and a half old, my family moved to Texas. We moved to College Station, Texas, which is also known as Aggie Land. I did not go to Texas A&M for college. I instead went to a place associated with that building. Does anyone recognize that building or those buildings? I'll give you a hint. Yes, exactly. It's Bobcat Village Apartments. I lived there for 2 years. I had a roommate who the only thing that we had in common was that we did not want to talk to our roommate. It was really fantastic. The only time that I ever really remember having a conversation with him was at the end of year one. He said, "So, you gonna move?" And I said, "No, I don't think so." And he said, "Okay, well, I'm just gonna go ahead and put in my request to be back in this apartment." And I said, "Sounds good." And that was pretty much it. It was really fantastic. It was a good working relationship. So I got my bachelor's in biochemistry from Texas State University in 2006. And then after I finished my bachelor's degree, I went to Texas A&M, where I got my PhD in biochemistry. And what I studied was this right here. I studied that plant. Okay, so now I've asked several questions. Does anyone know what leaf that plant is from? I'll give you some hints. One, you eat the fruit from this plant. That's one hint. Two, this fruit is a key part of a BLT sandwich. And the third hint, the proper name for this plant is Solanum lycopersicum. Really, my second hint was the best hint. It's a tomato. Absolutely right. I'm really disappointed nobody said a bacon tree, but if I could make up a bacon tree, I don't mean any disrespect, but I think that I wouldn't be working here because I think I'd be retired because I would be making a bacon tree. At any rate, so this is tomato. These are leaves from a tomato. And what I studied as a graduate student was basically how tomatoes and plants in general respond to bacterial pathogens. So if they, if a bacteria attacks them, what do? Obviously, they can't stand up and run away, so they have to fight it off. And so how do they fight it off? That's what I studied. It was really pretty fun and pretty interesting. So these are all things that you don't need to know in terms of like, is this going to be on the test? The outline of the shape of Wisconsin will not be on the test. Where I was born, it's not on the test. But where was J.J. Watt born? It's not going to be on the test. I'm being facetious there. Okay. So with that in mind, we've got a schedule. That's on our Canvas page. That day-to-day rundown that I showed previously, that's how I expect you to kind of navigate the semester. The one, to me, drawback of that is that it's a little bit cut and dry. It's pretty simple. It's bare bones. It's minimal. And then remember, as the semester progresses, you got to scroll through because the later dates are at the bottom of the page. One thing that I want you to notice about that is our class will meet for 16 weeks this semester. So that means if we're meeting 2 times a week on average for 16 weeks, that means we're only going to meet 32 times this semester. And that's just, on day one, that seems like, oh, that's an infinite amount of time. But on meeting number 28, it's going to be like, oh man, there's a lot of material that I need to prepare for the final. So, just kind of stay on top of things. Now, with that, I would like to go back to my PowerPoint for a second. And in that PowerPoint, I'm going to, okay. So our Canvas page has our day-to-day plan. All meetings are going to be online, including our exams. We'll have a mixture of synchronous and asynchronous meetings. And by that, I mean asynchronous. I will post a pre-recorded lecture and I'll say, hey, we're going to be doing this. And here's the next time that we're going to meet synchronously. So what that means is there will be some times that this 2 p.m. time, there might just be a series of questions for you to answer that accompany the pre-recorded lecture. Synchronous meetings, those are days that we're going to have activities. Now, with that in mind, every single time that there is a meeting, I'm going to record it and post it on my YouTube channel. And that's going to be up and that's going to be available for you to look at for however long you want to. At the beginning and at the end of every week, I'm going to attempt to put a little kind of summary of like on Mondays, here's what we're going to be doing this week. And then on Fridays, here are the important things that we covered. Here's good ideas of what to review in anticipation of exams and exam questions. So, is everyone good with that? Excellent. Excellent. Okay. Now, next up is communication. I will mostly communicate with y'all using the announcements tab on Canvas. So anytime I send out those announcements that are like, this is what's going on this upcoming week, those are going to be class-wide announcements on Canvas. So what that means is that if you you need to make sure that your announcements don't end up getting like filtered to the spam folder. In addition to that, I like to keep my announcements as simple and succinct as possible. And so don't expect like a 1-page long announcement from me saying like detailing everything that you need to do in the coming week or anything like that. Instead, expect something that's pretty short and brief. Because I'm just gonna like pepper you with like, here's 3 important, here's an important thing, here's an important thing, here's an important thing. Those would be 3 different announcements. They're 3 distinct important items. So I worked for somebody or worked with someone a few years ago who said that if an email is longer than a paragraph, then they don't want to have it, or it's better to have a conversation about that, a Zoom conversation. And so I kind of believe that as well. That's not to say if you send me a 2-paragraph long email that I'm going to ignore that. I'm not. I'm going to read it and respond to you. But some things like you can imagine if you're working out a complex idea or you want to describe a complex situation, sometimes it's easiest to do that in person or in a Zoom meeting as opposed to, here, I'm going to type this up. So announcements are how I'm going to correspond to you, and just make sure I'm going to go to spam tool or spam filter. So I'll do some tests of this a little bit later. Individual emails, if I send you one directly, that's not going to be super common. Usually I like to I like to send messages that are like, hey, I noticed on the first exam you got an 80 and the next one you got a 52 and then the next one you got a 25. Is there anything I can do to help out? So that's, that goes along with my idea of being like simple. Like I say, here's the problem, here's the thing that I've noticed, what can I do? And then I'll usually propose we meet for a Zoom meeting or something like that. I just like to, that's one of the reasons that I'll send an email. Or, if I notice that you've missed all of our synchronous meetings throughout the semester at the midway point or something like that, I'll email you and say, hey, I noticed that you missed these. Is everything okay? And I might even put in a, what's it called? I think previously it was called a pause alert. And that's just kind of a well check. It's a internal thing on the university or at the university that is a way of checking to see that, you know, everyone's okay or somebody is okay. If you miss a lot of class, that could be an indication that you're struggling with mental health at the moment and, you know, you're in maybe a dark time or something like that. And so that's one of the ways or one of the reasons you might get an email from me. Just making basically seeing if there's anything I can do to help you out. And if there's anything wrong, if there's not anything wrong, then just say, no, I'm good. So office hours, what I'm going to do is my blanket response to office hours is that I don't schedule them. But this semester I'm going to. The easiest way to arrange an appointment is just email me. And what I'm looking at when it comes to an email correspondence is, hey, Dr. Gray, or this would be like a good email. Hey, Dr. Gray, I've got some questions about our upcoming exam. Can you meet on Zoom at 1:30 p.m. on Friday the 5th? That's a good email to me. What would I consider kind of a bad email would be, hey, I need office hours. When can you do office hours? And I'm really just kind of looking for like, I want to treat you with respect and I want you to treat me with respect. And I think that one of the ways that that is conveyed is with how you know, form an email. And so emails from me, you'll always see a salutation. And, you know, thanks, yeah, salutation at the beginning and then a, basically, yeah, a greeting and a salutation. There we go. So what I'm going to do later today is a poll for office hours that work. I'm going to propose a couple that work for me, and then I'm just going to go with majority rules. I'm going to, you know, let's say Friday, 8 a.m., Friday, 11 a.m., and then Thursday, let's go with Wednesday, 12 p.m. And so I'm just going to survey everyone and say, you know, these are the three options. And whichever, you know, let's say this gets the first highest number of votes, this gets second. Then I'm going to pick these two. And I'm going to say my office hours are these. If those don't work for you, which inevitably some of them won't, then I'm just going to say, okay, well, I need you to propose anytime you have questions or need office hours, propose an email or sorry, write an email and propose a time to meet with me. Okay, so those are the biggest things. Like we're remote. Communication is obviously electronic. I'm glad to meet with you in Zoom pretty regularly. I'll send announcements out on a regular basis, make sure that the spam filter doesn't catch those emails and cycle them to spam. And so that's something that I'll just post like a quiz or something or a survey type of thing that says I sent a announcement at this time. Did you receive it? So that will be another thing just to confirm that everyone's getting my communications. Okay, next up is one of the biggest parts of this class is the grading. So we'll have quizzes and homework. One of the things that I want you to know about this is that, like I said, you don't have to buy anything for this class. So your quizzes and homework, what they're going to appear as is Canvas quizzes. I'm grouping these two things together. These are open note. And I'm grouping them together because I might refer to this as homework. I might refer to them as quizzes. I think for uniformity's sake, I'm going to say, you know, quiz number 1. But it's still an open note thing. These are just going to be questions that cover the material that we covered in that day. And basically what you're going to do is you're going to have 48 hours to answer, you know, 5-10 questions. And some of them will be short answer. Many of them will be multiple choice or multiple select. Now, with that said, you'll have 2 attempts. And of those 2 attempts, you will get the score. So that I'm going to say this is our first attempt. This is our second. In the first attempt, you will see the quiz that you submitted. You will be able to look at that and you'll see that, oh, I got a 6 out of 10. So you'll see question 1, 2, 3, 4, 5, 6. Here's what you answered. You will not be informed as to which ones are correct and incorrect. So, if there's 5 questions on here, this means that you got 3 of those 5 right. You will not be told prior to the due date what questions you got wrong. So what that means is you'll be told, okay, you got 2 wrong. Which 2? You don't know. You're going to have to look back at those questions and say, okay, well, let's see. This first question was this. Second question was this. I feel really good about question number 1. 2, I kind of flipped a coin. Okay, so now on my second attempt, I'm going to resubmit. I'm going to change my answer for question 2. And so this will be like an opportunity to reflect and study those questions. I'm expecting, since these are open note and you have the chance, you get this grade feedback, that on your second attempt, I'm expecting most people to get 10 out of 10. I understand it's inevitable that, people will run out of time. They won't have the chance to do that second one, or they'll forget to, or something like that. So as a result of that, 2 are going to be dropped by the end of, or at the end of the semester. 2 of those questions will be, or 2 of those quizzes will be dropped. Now, when it comes to the 2 attempts, let's say that you get a 6 out of 10 on the first one and a 10 out of 10 on the second one. Well, then the 10 out of 10 is what is going to be used for your grade calculation. These are going to be worth a grand total of 15%. Okay. So what this means is you're going to look at the grade book, and the grade book will have a quiz section. And that will have quizzes 1, 2, 3. I would bet that over the course of the semester, there's going to be, 20 quizzes, roughly. There might be more, might be less. It's not going to be like, oh, there's actually 60. If it's more, it's going to be like 26 or something like that. And if it's less, it's going to be about 15. So I'm going to say roughly 20. Of those 20, the highest 18 are going to be averaged together, and that is going to be worth 15% of your grade. Okay. Now, you have a final exam, which we've already talked about the date. That's a cumulative final exam. And then we have activities and polls, and that will be for 10% of your grade. Now, the trickiest part about, okay, so before I proceed, can everyone add to the chat the thumbs up emoji as a way of saying, yes, I understand? Okay, we got 1. Thumb 2, 3. Good. Here we got 5, 6, 7, 8, 9, 10. 11 is mine. Okay. 12, 13. Okay, we got 13. We need 20. 14. We're almost there. 15. I think if we get to 16, then I'm just going to go ahead and proceed. 16. We did it. We did it. Thumbs up emoji to that. Okay. So I wanted to make sure everyone was good on that, because this next part is a little bit complicated. And I want everyone to not lose sight of, or not get distracted by the complexity of this. So I had to drink some water to get all geared up for this. Okay, so we will have a total of 5 exams. Those exams will be administered by on Proctorio. I'm going to give like a sample quiz where Proctorio is used just to make sure that you have your exam set up good to go. Proctorio has to be done. Proctorio cannot be done on an iPhone or an Android, and I'm not sure. I don't think that it can be done on a tablet. I could be wrong. But a laptop or a desktop computer is your best bet for Proctorio. That's something that we'll get to a little bit later on. But what I really want to focus on right now is you're going to have a total of 5 exams. So what that means is that the semester's material is broken down into basically 5 modules, we'll call them. Now, when it comes to the grade book, that's the important thing that I want you to know about. Whenever I look at the grade book, I have basically groups. Exam 1 is a group. Exam 2 is a group. Exam 3 is a group, or category, you could call them. Exam 4. And then last but not least is exam 5. Okay. So I believe that it is the 17th of September. You'll come in, well, you'll get on Proctorio, and you'll take your first exam. And that exam will have nothing but Module 1 material. So I'm going to label this as M1. So it's only the stuff that we've covered from today till like the 14th or whatever it is. So let's say hypothetically that on that exam, you get a 55. And I like to start with a low number just because I feel like that's from my experience pretty common where, you know, I'll go into a new class, a new professor, and I'm like, how do you test? Oh, okay, this is, I learn a lot in that first exam period. I learn about the questions that are asked and the way that I prepared for it, and I'll be like, ooh, I totally neglected this stuff. I need to focus on that. Okay, so that's exam number 1. Then, so this is 9/17, so September 17th. Then I believe that it's on October 8th, maybe, give or take a day, 10/8, you're going to have exam 2. That exam will be roughly 30% module 1 stuff and roughly 70% module 2 stuff. So exam 2 is going to be cumulative from what you've covered so far. Exam 3, which I think is 10/29. Again, I could be off on my dates here, and I'm going to stop at this point. So exam number 3 is going to be module 2 and module 3. And it's going to be about 30% module 2 and about 70% module 3. So exam number 3 won't have any of this first block, this first module of material, but it will have modules 2 and 3 on it. Okay, now let's go ahead and fill in our blanks here. Let's say on exam number 2, you get a 78. You get a 78 because one thing that you did before you took exam 2 was you looked back at module 1 stuff. You said, whenever I took exam number 1, I had questions about drug discovery. So I'm going to review and revisit that drug discovery stuff. And whenever I do that, oh, I've learned it better. Great. You get a 78 on that. And so I'll put that into the category for exam 2. Then exam number 3. Well, exam number 3, let's say that again, exam 3 is over module 2 and module 3. So you really study that really clearly and you do better. Let's say that you get a 95 on this. Okay, so I'm going to put that in my category, exam 3. So you get a 95 on that. Okay, now we're going to go back for just a second. I got to change my ink coloring and go with green. Now, if we think about times that you've seen material for, or module 1 material, you saw it on exam number 1 and exam number 2. So what I'm going to do is in this category for exam 1, I've got the places where I saw module 1 material. And that was on exam 1 where I got a 55, and then on exam 2 where I got a 78. Okay. Now, exam 2, that was covering, I'm going to change my color again. That was covering module 2 and module 3. So now we have a second place where we saw module 2 material, and that was on exam 3. So that's where I got a 95. I'm going to put that 95 in right here. Okay. And then last but not least, I'm going to jump ahead a little bit here. Your final exam. This is a cumulative exam. This exam is going to have a little bit of module 1, 2, 3, 4, and 5. So everything that we cover over the course of the semester is going to be on the final exam. And let's say hypothetically that over the course of the semester, you learned, okay, here's the types of questions that Dr. Gray likes to answer or to ask. Here's the material that Dr. Gray asked about. I'm going to comb through that and make sure that I'm in good shape. And by that time, this rolls around, and let's say that you get a 91 on the final exam. Now, let's go back to E1 here. E1 was an exam over module 1. So we saw module 1 on exam 1, and we saw it on exam 2, which is why we have a 55 and a 78. Now, can anyone add to the chat where else we saw module 1? If we saw it on exams 1 and 2, where else did we see it? The final, exactly. So to reflect that, E1, you're going to have the 91 added here. Okay. Now, what I'm going to do for each of this, like this E1 category, that's worth 15% of your grade. In Canvas, I can set a rule to a grade book, or I can set a rule in the grade book. And that rule that I'm going to set is drop to lowest. And so then what you're going to see, or this is the tricky part about Canvas for what you see versus what I see. What I see is perfectly clear. For you, it's a little bit less slow. So what matters is that when you look on Canvas and when your grade is calculated, this 55 and this 78 are not included in the grade calculation. But instead, only this 91 is for, or sorry, not 15%, but instead 12% for that E1 group. Now, can everybody who understands, or, okay, now, does that make sense? Okay, I've got one yes. Can we get a second? Perfect. Okay. So that's what happens when your final is higher than your other opportunities for that material. The final average or the final score that you got kind of brings everything else up. It drops grades that are lower. Now, if we look at E2, that's where the situation's a little bit different. E2 was over module 1, or sorry, module, the focus of E2, the primary focus was module 2 stuff. Where have you seen module 2 stuff? Well, you saw it on exam 2, where you got a 78, and then exam 3, where you got a 95. The last place that you saw M2 was on the final where you got a 91. So you're going to have 3 grades that are under E2. You have the 78 from exam 2, the 95 from exam 3, and the final of 91. I'm going to set the exact same rule, drop the 2 lowest. Well, the final is one of the 2 lowest. The 78 is the other lowest grade. So the 2 grades that are dropped are not always going to be the regular midterm exams. They might be the final, they might not be the final. So this 95 is going to be used for calculating your final exam or your final grade, I'm sorry. So this group is going to be worth 12%. Then you'd go on, you'd say, okay, well, you look at what was earned on exam 3, what was earned on exam 4, and what was earned on exam 5, compare all of those to the final. And the intent here is, okay, before I proceed, I'm looking for, does anyone have any questions on that? And maybe another way of phrasing that is that clear? I know it's a little bit tricky, but if you understand, could you add the celebration emoji? The ta-da, I believe is what it shows up as. If you understand, add that.
No, that made sense at that time.
Okay, nobody's okay, wait. I got one vouching for that. If you would like me to kind of write out some scores, example scores, if you want example grades for everything and how that would look, use the face holding back tears emoji, that one. I'm gonna wait 20 seconds. I'm getting lots of party emojis, so that's a good indicator. Okay. Okay. If you want some clarity on this, please don't hesitate to contact me. The real intent of this is to ensure you have an opportunity to basically raise your grade over the course of the semester. You know, if you do bad on the first exam, that doesn't doom you to fail the class. If you do bad on the second exam, if you do bad on the first 2 exams, these things don't doom you for the entire semester. Now, I'll say as a cautionary tale, there's always someone who sees this and they think, aha, so what you're saying is that I can skip every single exam, then roll up to the final, ace the final, and then I've aced the class. Is that what you're saying? And call me a, well, I don't know, just a boring person, but no, that's not what I'm saying at all. And that's a horrible idea. That's missing the point of learning. That's not the point. The point is that you take the first exam. If you don't do well, review that exam, study that exam, learn from that exam, and do better the next time you see that material. What I would love to see happen is everyone's grades gradually increase because they're learning different things. They're learning new things every time they review the material. So that's that. Now, exams, initially, we're going to start out with everything being administered on Proctorio. The exams or the questions will be a mixture of multiple choice and multiple answer questions. Now, the way that multiple answer questions work is it would be something like, let's go with select all true statements about Michael Jordan. And the answer options would be let's go with played minor league baseball. played for the Lakers. Let's say went to UNC, and then let's see that I need two more. Owns a home, or actually is from North Carolina, is an only child. Now, if you don't know any of these questions or any of these answers, well, get ready. Okay, so this would be like a 2-point question. And if you said, okay, well, he played minor league baseball for the Birmingham Barons, correct. And you said, oh yeah, he played basketball. He played for the Lakers, and you marked that. Okay. Then you said, oh, well, he went to UNC and he is an only child. Okay. So these would be the 4 answers that you selected. You said, these are all 4 true statements about Michael Jordan. Okay. Here's how Canvas would grade that. They would say, okay, played minor league baseball. Check. Played for the Lakers. X. Went to UNC. Check. Is an only child. X. Now, does anyone care, or would somebody add to the chat how many points you would get from this? Or how many points you think you would get from this question based on these answers? Correct. That is worth 0 points, because although you answered some things that are correct correctly, you answered some that are incorrect incorrectly. I always think about it as 1 step forward, 1 step backward. There's not going to be negative points or anything like that, but I think that these multiple answer questions are really kind of how well do you know this information? And I think that there's kind of a, I don't know if I would say a strategy, but if you're feeling very bold about it, then you go ahead and you answer all the ones. If you are not too sure on the topic, then you say, okay, well, these 2 things I know are 100% true. In which case, let's go back for a second, and let's say I'm going to erase, erase, erase. Okay, so let's say that, okay, so let's say that this was the marking scheme that you came up with. The X's indicate that you marked it. This would be, this is correct. He went to UNC and he is from UNC. The thing is, you didn't mark that one. So of the 2 possible points, you got 2/3 of those. So you would get like 1.3 points from that question, because you marked part of it right, but not all of it. You did not select all of the right answers for that. Okay. My plan is for the exams to all be administered on Proctorio. Our exam time, or our class, is 2 p.m. to 3:20. And what I always like to do is give a little bit of a buffer. I'll say the exam will open at roughly 1:30 p.m. and it will be open until around 4 p.m. Now, within that time frame, you have 1 hour and 20 minutes to complete it. So what that means is you have 2.5 hours to complete a 1 hour and 20-minute exam. So if you want to start at 3 o'clock or something like that, by all means go for it. You're limiting yourself because the exam will close at 4 p.m. exactly. If you need accommodations, please contact me so that I can make sure that I add in that time so that you get the accommodations that you are afforded. So please contact me if that's a part of the deal, and I will add that. Other than that, though, these are pretty much the important parts about how this class is going to function and what you should kind of anticipate. Does anyone have any questions about any of that? Okay, well, this is, for all intents and purposes, that is basically the syllabus in long form video. or long video form. And I say video because it's being recorded. Now, with that in mind, we're going to go ahead and proceed to go ahead and get started on our new material. So, okay. What we're going to talk about today is the process of development of drugs. And these would be therapies or therapeutic drugs, chemotherapies, or basically any other drug for that matter. Now, when we talk about drug design, what we really need to be most familiar with is the target protein. So that's the part within a cell that carries out a particular function. And when it comes to drugs, drugs are molecules that interact with a target protein. So I always think about this, I always like to use examples in parallels. So, you know, I think about like a car or something like that. If you have a steering wheel is similar to a protein within a cell. The steering wheel carries out a function. Now, that steering wheel in a car, if you break it in some form or fashion, or you prevent it from working, does that mean that that cell or that car, for this example, does that car die immediately? No, it doesn't. I would argue that no, it doesn't. Instead, that car or that cell is going to carry out its specific function, but it's going to be kind of limited. And the way that it's limited is that car can't turn. So inevitably, is it going to run into a wall or run into something that it can't basically move around? Yeah, it's going to run into the ocean or something like that. So this is the molecule is a way of basically disabling the function of a protein. So the first thing that we need to do is we need to find a molecule that interacts with a target protein in a specific way. Next, what we want to do is we want to apply that in cells, which is known as an in vitro setting or referred to as an in vitro setting. So sorry about that. It's going to be done in cells or in vitro and in animals, also known as in vivo. If you've ever heard this word vivo, this is another way of saying it's basically alive. So this is why, you know, animal testing is done effectively or basically, because you basically progress through. Whenever a molecule is found that carries out a particular function, well, then you say, okay, cool, that we've isolated this protein and we've isolated this molecule and they interact. Great. Now let's see, does this interaction take place in vitro when you have an isolated cell? Does that interaction take place? Oh, it does. Great. Okay. What about whenever we inject that molecule into a living system or into a living organism? And there's kind of a hierarchy of sorts. There's mice, rabbits, all the furry little animals are selected and basically progress through. And then you get into primates like monkeys. And then if everything's looking good and this is effective in those, then we're going to move on to testing it in humans. Now, these first three steps are pretty standard. The FDA reviews all of the data from those first three steps. Were those animal methods effective? Were the in-cell methods effective? And then did we go through mice, rabbits, and monkeys? And then the FDA says, okay, is there enough scientific evidence that was collected and analyzed correctly? Okay, so this part right here. One, if the evidence was collected and analyzed correctly. Data analysis can be very challenging. You can analyze data and come to an incorrect conclusion. This is kind of a question of correlation and causation. So if you say, oh, well, you know what? I go to the gym every day and I've been lifting weights a lot and I've noticed that I'm getting stronger. And I think what it is it's because before I go to the gym, I watch an episode of SpongeBob SquarePants. So I think, everyone should watch an episode of SpongeBob SquarePants. And then they're going to be like super strong. Well, that's identifying, okay, I'm getting stronger, and then attributing it to watching SpongeBob SquarePants. It's actually kind of disconnected from the actual outcome or the observed outcome. So this will determine if the drug is safe and effective for humans. That's the role of the FDA, or the Food and Drug Administration. Now, the pharmaceutical company and Food and Drug Administration continue to monitor the safe and effectiveness of commercially available drugs. There have been instances when the drug was deemed to be dangerous and pulled off the market. So, as an example, valdecoxib. This is a drug, if I remember correctly, this was one of the drugs that was a basically it was a anti-inflammatory drug. Or I'll say it's an anti-inflammation drug. So if you have a fever, you would take this instead of aspirin or ibuprofen, because it would stop the formation of the molecules that produce an elevated temperature in your body. But if it wasn't doing it, then it was removed from the market. Now, when it comes to this process of drug development, you've got the discovery and development. You've got your preclinical research, your clinical development. So those first three are highlighted right here. You've got the FDA review, and then post-market monitoring. So when this drug is actually out there, you're still keeping tabs on it. Are there instances where the drug is not being administered? Why? So this is the entire process in a nutshell. What we're going to be focusing on is, where do we even start? So we have a couple of different or the phases of drug discovery are target discovery, target validation, lead generation and refinement, and preclinical development. Okay, so the idea here is, for this first phase, is to find all of the target molecules. So molecules that interact with a protein. So what you have is the protein, and then you screen and say, what are all of the molecules that could possibly interact with this protein? So that's your target discovery. Then your target validation is where you say, all right, well, of those, 250 targets, which of those do not actually do this interaction? So this would be kind of like, let's say, this is kind of, let's see, I'm trying to think of a good example. If you got a phone book, I don't know if anyone has a phone book anymore, but if you got a phone book and you said, okay, well, I am looking for my friend John Johnson in the phone book. So the first thing that I'm going to do is I'm going to find all the people whose last name is Johnson. And I'm going to get all those numbers. Those are my targets. The next thing that I'm going to do is I'm going to validate some of those targets. And I'm just going to call every single one of them and be like, is John there? Is John there? Is John there? Is John there? And I'm going to narrow it down to, let's say 15 out of 250. Then, after each one of those conversations where you said, is John there? And the answer was yes, and you hung up, you can call those back. Those 15 targets you can revisit. And that's where you have this lead generation where you've purified these molecules. You generate and purify these molecules, and then you call and say, hey, is John there? Oh yeah, this is Joel. And then if the answer is, oh, hey, Joel, Yeah, you're my friend. Okay, cool. That's my point where I have a molecule that's been selected. I have a family, or I have a molecule that has it interacts with my protein. Then what I'm going to do is go on to this preclinical development where I take my drug and I administer it to animals, mice, rabbits, monkeys, and see does this actually like progress and does it function? Now, what are drug targets? Drug targets are typically proteins in the human body that are involved in causing the disease or can help alleviate the disease. Now, an example of a drug target. The common antibiotic, amoxicillin, which I'm pretty sure that almost everyone in here who has ever had an ear infection has taken amoxicillin. This targets the penicillin binding protein in bacteria. Okay, so the drug here is amoxicillin. The target protein is the penicillin binding protein. Now, the desired effect of taking this drug and applying it to this target is that the bacteria is going to die because the cell wall gets damaged by this drug binding to the target protein. Okay, so when we're talking about drug design, and we will proceed through this over the course of the semester, think about what is the drug and what is the target. So these two interacting partners, I like to use that phrase, interacting partners. So you could say the interacting partners are a drug plus a protein, or say drug and protein. Okay. Now, how do we know that we have the right target? So target validation helps us verify which protein is involved in a disease. So if we go back for just a tad, we have this bacteria that is causing some sort of problem, whether that's causing an ear infection in the person or another type of infection. We have pinpointed a protein that basically keeps that bacteria from dying. Now, there are several ways we can determine if a protein is involved in the disease. One way is we can do what's called genetic manipulation. We can change how much protein is in the cell by turning on or turning off a gene. So what that means is we can try to grow bacteria with and without that amoxicillin. I'm going to call it ABP. I'm going to jump back. All right, sorry, PBP. Let me erase this for a second. PBP. When you grow this bacteria without the penicillin binding protein, it doesn't live. Okay, cool. That target protein is a good target to go after because when you knock it out, what happens to the bacteria? The bacteria dies. So I'll just say bacteria dies. If the protein is not made because a gene is turned off and that causes the diseases, then we validated that the protein affects the disease is a potential target. Another way is with molecules known as antibodies. Antibodies can bind to the protein and prevent it from working in a cell. If the protein affects the disease, then blocking the activity may either cure or cause the disease in the cell. Then last but not least is a process known as chemical genomics. We can use chemicals known to be involved in the disease and see if they bind to that protein. If the chemicals do bind to a protein and change the protein's activity in the cell, then it can be validated as a target. So what we can do is basically identify a target, and we can either manipulate it genetically, we can use an antibody to kill it, or we can use some sort of chemical compound to kill it. So essentially, when you're studying a bacteria, for instance, you say, aha, PBP, the penicillin binding protein. I think that is what amoxicillin binds. First thing is grow bacteria with that protein present, so the kind of native or wild or regular bacteria, and then grow it without that protein and see the effect of that. Next up is to use an antibody to basically block that PBP from functioning. So anytime you have a target, figure out a way to basically knock it out. And that's with an antibody, a chemical, or genetics. Those are your three ways to validate the target that you're looking at. Now that we have a target, how do we actually design a drug? Now we can use computers to look at how chemicals bind to proteins. This will be our main focus for our class. We'll visualize how chemicals interact with and bind to proteins. Now, in order to do that, we have to learn some organic chemistry and biochemistry before we can understand how chemicals interact with proteins. So if any of this has been daunting, I want you to know that we're going to be easing through this. So that phrase organic chemistry or biochemistry might be intimidating, but I'm going to give you the tools that you need to know and the basic information that you need in order to work through this. Now, organic chemistry and drug design. Organic chemistry is a field of chemistry that's really focusing on compounds that are based on carbon. So another way, carbon-based. Now, whenever you hear, organic strawberries, well, that has to do with the industrial process and the agricultural process by which those plants are grown or those strawberries are grown. That's different from this. Organic in that sense means not using pesticides or herbicides. Well, organic chemistry is a little bit different. Organic chemistry allows us to understand what interactions a drug can form with the target protein through or by observing the chemical composition of the drug. Interactions are how chemicals attract or repel one another. An example of a favorable interaction is that sodium is attracted to the polar water molecules. Now, an example that's a favorable interaction. So this is ion plus polar molecule. And this is specifically a positive ion. An example of an unfavorable interaction would be a sodium ion and a potassium ion, which will repel one another and not interact. So this is a positive ion and a positive ion. Now, a positive ion and a negative ion will attract one another, but a positive and a positive will repel. Now, an example, prednisone has oxygen atoms that will allow it to be slightly soluble in water. This is a picture of prednisone. And these red balls right here are all oxygen atoms. Everything else is carbon and hydrogens. Okay. Now, biochemistry, or the field of biochemistry, allows us to study the target protein, the interactions of the protein with the drug, and how the target protein is involved in disease. So here what we have is an example of structures of proteins. So biochemistry as a field and biochemistry related to drug design is basically a field where you try to find out what something looks like. Because if you have a sense of what that thing looks like, then you can develop like a plan of attack. I always think about it as I tend to think about this almost like what's the story of the three little pigs. You have an idea of what these houses look like. You've got the house made out of straw. Okay, well, I'm going to blow that over with a leaf blower. Then you got the one that's built out of sticks. Okay, I got to get 2 leaf blowers to blow that over. Then last but not least, we got the house built out of bricks. Now, is that house that's built out of bricks, is that completely indestructible? No, it's not. It has a special shape. Does that house that's made out of bricks have any form of weakness? Well, in the story of the Three Little Pigs, the answer is no, it doesn't have any weaknesses. You know, the big bad wolf just was trying to knock it over for the longest time, couldn't do it. Well, but that's not realistic. I mean, that house did have weaknesses. And if the big bad wolf was critical and analytical, what he'd do is or they would say, you know what? I've tried blowing this house down for an hour, and that's not going to work. I think what I need to do is I need to find one of the weaknesses. I can see the structure's made out of bricks, and there's mortar that's connecting all those bricks. I think what I'm going to do is get 100 leaf blowers. And what I'm going to do is basically put those into the windows of the house. I'm going to put them into the windows, and then I'm going to seal the house off so that that air basically goes in and has nowhere to go except to break that house open. That's if, and I just decided my $1 million idea, I'm going to rewrite Three Little Pigs with where the big bad wolf wins. But essentially, that's what this is. The more that you know about the structure of a protein, the more that you know about the structure of the third little piggy's house, the more you know about the weaknesses, the more you know about where it can be broken down and what can you do with it. So that's what biochemistry, or one of the parts of biochemistry, is after. What does something look like? If you know what something looks like, then you know its weaknesses. Like, you know, I'm trying to think of another good example. I think I've tapped out with a fantastic Three Little Pigs one, though. So over the course of this semester, that's what we're going to be focusing on. We're going to be focusing on drug design. We're going to be talking about drug design and biochemistry of drug design. We're going to start out learning a little bit about organic chemistry. The organic chemistry that you need to know, I'll give you everything that you need to know. There's not going to be anything that's like too daunting, because we're going to work through this pretty deliberately. And anytime you have any sort of question, please don't hesitate to contact me. With that said, I'm going to go ahead and stop. I'm going to stop my recording.